My talk is going to focus on the immunogenicity of therapeutic antibodies and attempts to avoid that. Many therapeutic antibodies are given long-term, and consequently, because they have foreign elements in them, they can elicit immunogenicity. And attempts have been made over the years to minimize that immunogenicity, not least of which have been engineering attempts through humanization. But despite the best efforts, immunogenicity still remains a problem for some antibodies for some people. And so it would be very useful to find a way to avoid that. We all know that our own immune system is tolerant of our own immunoglobulins, and so one route to try to avoid immunogenicity is to harness tolerance mechanisms so that the antibody that goes in therapeutically might be able to elicit tolerance to itself. And that's where I want to discuss work we've been doing and where we've got to. So you all know about humanization, and it's a technology developed by Greg Winter in which the hypervariable regions, the loops of antibody that interact with antigen, are transplanted onto a framework which is human and largely then acceptable to most recipients. You'll see in purple there the areas that come from the rodent, and those loops can contribute to immunogenic peptides. Generally speaking, for short courses of treatment, humanization has been very useful. But when it comes to long-term treatment, many patients eventually do make responses to these purple areas, these hypervariable regions, and not least of which you all know about anti-TNF, and I think I've heard statements saying that up to 50% of people do make anti-idiotype responses to the loops or anti-TNF antibody, and for some people that means stopping and changing therapy. So it is an issue despite humanization, or even what are called fully humanized antibodies. The antibody I'm going to concentrate on is Campath. It's now known as alemtuzumab and Lemtrada, and it's licensed for treatment of multiple sclerosis. And even though in multiple sclerosis it's only used in a couple of courses, after the second short course, about 70% of patients will have made an anti-idiotype response to it. So if ever later retreatment is considered, there is a possibility that sensitization will in some way impede the future therapy. And this may be true of a number of other antibodies. So humanization, I'm saying, has not been absolutely sufficient, although it's been very valuable. The approach that we've adopted is one that actually only old people like myself could ever think about. It came from the work of Chiller and Weigel before I even started my PhD, and it's been very influential in a lot of my thinking about tolerance. What they described was the fact that a mouse that was given a deaggregated form of human immunoglobulin that is there as monomer would become tolerant to that immunoglobulin, even though the immunoglobulin was foreign. And tolerance was measured then by immunizing later or challenging with the gamma globulin in an aggregated form. That aggregate is usually very immunogenic, and what they found was pre-treatment with the deaggregated immunoglobulin tolerized to the aggregated. People like Polly Matzinger would now interpret that saying, well, the deaggregated immunoglobulin posed no danger, gave no innate immune signals, and the immune system's not geared to respond to something that has no danger signals. Anyway, the issue here is that you have a mature immune system in a mouse that has been rendered tolerant through the way the antigen is perceived. And we have tried to apply that principle to inducing tolerance to therapeutic antibodies. we reasoned that the aggregate that Chiller and Weigel were dealing with would probably be the equivalent of antibody binding to a cell surface, as you see in the lower part of the diagram. And if antibody didn't bind, or was binding monomerically through a receptor, it might induce tolerance. But if it was binding to a cell surface, it was an aggregate, and it might create danger signals because of attracting Fc receptor-bearing cells, attracting complement, and eliciting inflammation. So the idea was, if you could render your antibody monomeric and not binding to the cell for a limited period, then you might create tolerance to the therapeutic. And this is what all the work I'm going to discuss with you later is going to deal with. So in all the mouse studies I'm now going to talk about, what we've done is to create a mouse that is transgenic for the target of the Campath antibody, that's CD52. It's expressing CD52 on its own T lymphocytes. And we're injecting Campath, the humanized form of the anti-CD52, into these mice. So it's completely foreign. It's a human immunoglobulin into mice, and it's binding to the surface of the T cells of these mice and is able to kill the T cells, is able to use complement, is able to attract Fc receptor-bearing cells like macrophages and so on, and create inflammation. And what I'm going to show you is that the Campath is very immunogenic in mice that have CD52. And in fact, if we avoid the antibody binding to the cell surface, we can, even by creating a single amino acid mutation in the loops, we can make that antibody tolerogenic. In other words, pre-treating the animal with that mutant gives tolerance to the therapeutic antibody given later. So let's start with that. Of course, that's a much bigger challenge than humanized antibody into a human because, as I said, humanized antibodies, the only big immunogens that are generated are derived based on the loops. Maybe some allotypes, but the loops are the issue. So let's look at what we do to create a mutant. What we initially did was to take the antibody, look at the CDRs, and replace each CDR one by one in turn with a neutral CDR, a loop, and try to identify a CDR that was crucial. And we found that CDR2 of the heavy chain was crucial. When substituted, the antibody lost binding. So that CDR is very short, and we could then go into that CDR and interrogate various mutants. And we found that we created a number of mutants. We called them mutant 1, mutant 2, mutant 3, single mutants, or double mutants. And binding assays indicated to us that some of these mutants bound very poorly in solid phase assays to the antigen. You see the original Campath bound very well when titrated in red. One of the mutants still had some binding, and then we had some mutants down below that had limited binding capacity. But otherwise, the protein was pretty well the same as Campath. Because it could no longer bind and no longer had the potential to generate danger signals, the question was, would it be tolerogenic, immunogenic, or what? So here's a sort of protocol. We take the animal that's transgenic for human CD52, and we inject into it two doses of either the Campath. We predict that should be very immunogenic, very foreign to the mouse, or a control antibody, human CD4, which won't bind, an antibody to human CD4, which won't bind to the mouse T cells at all. It will just behave as a monomer. But it won't be able to tolerize to the idiotype of the Campath antibody. It will only tolerize to its own idiotype, to its own loops. And then we've got these various mutants. And we come along 3 weeks later, and we simply ask, what was the immunogenicity of all those different mutants? And what you'll see is Campath would be very immunogenic, and all the others would be poorly immunogenic, as predicted. So although foreign, limited mutations, knocking out binding, and therefore knocking out aggregation, or surrogate aggregation and danger signals, lose immunogenicity for the therapeutic protein. But of course, therapeutically, that's no value to us because the antibody is not going to do anything useful because it's not binding to the target that we want to kill. So we ask a two-stage approach. Can we pre-treat with the construct which is non-immunogenic and then come back later with the therapeutic? And do we, as a consequence, get benefit for extended use of the therapeutic? In other words, if we tolerize to most of the protein, apart from that one amino acid area where the mutations occurred, would our therapeutic be more effective? So here's a protocol. We take one of the mutants, mutant 3, give a couple of large doses to get high zone tolerance. That would tolerize, we think, the T cells and the B cells, according to Chiller and Weigel. And then we give loads of injections of the therapeutic and analyze whether we got an antibody response to the therapeutic in that course. And what you'll see here is that pre-treatment with the mutant basically avoided us getting an antiglobulin response after multiple injections of the therapeutic. So giving something that didn't aggregate, that didn't create danger signals, did tolerize the therapeutic. Well, of course, that's exciting, and you could do that for a number of therapeutic antibodies. You could make mutants. But the reality is that no commercial organization will ever want to sell 2 antibodies to get the benefit of one, because they like to make profits. And then they have to go through all the development to show that the first one was safe and so on, and didn't induce benefit in its own right. The principle, we think, is relevant because we've gone back to patients with multiple sclerosis in collaboration with Alistair Coles and Cumston, the neurologist. And what you'll see here is studies where they've taken the mutant there in yellow, and if they pre-treat patients with the mutant and then give them Lemtrada, the first course of antibody, after the first course of 5 injections, they can't measure really much in the way of antibodies against the therapeutic Campath given a few days later. If they are pre-treated with no mutant, some of the patients respond on the first course, but when it comes to 2 injections a year later, the patients who responded, the patients who had no mutant to start with, 73% of them now responded, many of them with high titers. But those that had the mutant, about 21% responded, and you'll see here the responses on average were very low. Of course, we haven't got data from more injections later, simply because the patient's not given them. But the reality is that we do think that this would work in man. So as I've told you, it would work if anyone ever made the 2 mutants and sold them as a package, but that's not going to happen. So what I'm going to do in the last part of my talk is just concentrate on whether we can do this as a one-step maneuver. The idea would be here that the antibody you inject would start off not being able to bind to the cell, to its target, for some period of time, and only after tolerance has been induced would the antibody expose its binding capacity, bind to the target, create the aggregate, and create the danger signals. But that would be too late to elicit an immune response. That would be the principle. And to do that, we'd put an obstructive element into the antibody. We've engineered it in, so it's covalently associated through a linker. I'll just show you here. A flexible linker at the N-terminus of the light chain. So, and what we have in there is the mimetope. It's a structure determined by Jeff Hale, the amino acid sequence that is actually equivalent to the antigen. So that's held in there by the linker in both antibody combining sites. And the idea is that when we inject this, the antibody can't bind, but over time, the variable regions may become exposed, the binding sites may become exposed, bind to the target, and then over time, antibody accumulates on the target, and eventually you will get enough accumulating to then kill the cell. And we would predict that, would hope that that would happen at a time after tolerance had been induced. So that's the principle we were trying to adopt. We made various constructs, some as controls. Here you'll see on the top the wild-type antibody. The one I'm going to call the mimetope IgG1 is the one that's got the mimetope of the antigen in it. We've got a flu peptide covalently bound in there as a mimetope, a flu peptide bound into the binding site as a control. Also, to diminish danger signals coming through attracting effector cells and complement, we created a form of Campath that has had the sugar removed. And we've even created a form of this mimetope binding antibody that's, so this one now can't bind to the cell surface, nor can it attract effector function. And we've got a control there with the flu peptide. And we're going to be looking at the ability of all of these constructs to bind, to deplete, and to be immunogenic or tolerogenic. And our prediction would be that the one that would create the least danger signals would, of course, be the most tolerogenic. The one that would not be able to aggregate and create these danger signals. So this is basically what we've done. Again, we've had binding assays that tell us that our mimetope binding IgG1 binds very poorly, as does the aglycosal IgG1, binds very poorly. Putting in the flu peptide on a covalent with the linker does in fact impede its binding activity somewhat, but it still binds very well and can, by various binding assays, by plasma and resonance, we can't really detect a difference to control. So let's now look at what happens with binding in vivo. And these are data which are the fluorescent index of peripheral blood lymphocytes. The higher the value, the more antibody has bound. And we're looking 3 hours after injection of these various constructs. And what you'll see is the antibodies we expect to bind very well by 3 hours have really bound maximally to the cells. But in the red squares on the top there, you'll see that the two constructs with their binding site blocked have not bound very well at that time compared to the control right on the very left-hand side. And with spleen cells, they're almost not binding in a way that the untreated control. And if you look at their ability to deplete, you'll see again on the right-hand side, the two constructs with their antibody binding site blocked are not depleting CD3 cells, either in the blood or the spleen at 3 hours. So we think not only do they not bind in vitro, but even in the short term in vivo. And now if we look at immunogenicity, you'll see that the wild-type antibody after injection into the transgenic mouse is very immunogenic. That would be the group with the rectangular red block. They're the wild-type antibodies. But if you look at the ones carrying the mimetope, you'll see that they're poorly immunogenic. But I think of great interest is the fact that if you knock out not only binding, but also the ability to generate danger signals, you find that this antibody, purely monovalent, totally unable to attract effector signals, that is absolutely not immunogenic. So that's taught us really that we can knock out immunogenicity, but of course we knock out effector function. That's a bit unfortunate if you want to kill cells. When it comes to tolerogenicity, the same thing is pretty well true. If you pre-treat with a tolerigen, and then you come back with many injections of the therapeutic, you'll see that these antibodies have produced a major reduction in the host response against the therapeutic. The pre-tolerization, sorry, the constructs are not immunogenic for themselves. and not immunogenic for the therapeutic either. That doesn't have the blocking agent inside its binding site. But it's not absolute. The only construct that's absolutely non-immunogenic is the aglycosylated form that can't attract inflammatory events as well as, and cannot bind to the antigen as well. So I just want to go on to say that the aglycosylated one is completely tolerogenic at this big dose, 500 micrograms per annum. Right, so now we think we, in principle, could construct an antibody that doesn't bind to its target initially, but binds to its target later, and then avoids a great deal of immunogenicity. In animals, Chiller and Weigel predicted it would take about 6 days to tolerize the B cells, about 3 days to tolerize the T cells, and in their case, they were measuring the thymus. What we find here is if we take the absolute toleragen, the aglycosyl mimetope containing antibody, and we tolerize, go through the treatment of tolerizing, and then challenge, what we find is that if we challenge at 3 hours, we haven't got tolerance. We challenge at 8 hours, you'll see we haven't got tolerance. But by 24 hours, we've got quite a reduction, and by 3 days, we've got about a 30-fold reduction in immune response to the therapeutic, as measured, a challenge with the therapeutic. That's the original Campath. And by 14 days, tolerance is absolute. So the harsh reality is that you need, to get absolute tolerance, you need a good amount of time for tolerance to occur in the whole immune system. But probably by 3 days, you've probably got at least a 30-fold reduction in the potential to make an immune response. If we try to translate that from mouse to human, then in human, we're not dealing with something that's completely foreign. We're dealing with something that only has those idiotypes as foreign. So we might be able to say, even though we haven't been absolutely successful in the mouse to tolerize the completely foreign antigen, had we just had the antibody being what you might call murinized and the loops being foreign, I suspect we would have probably seen that we have complete tolerance to that, or complete inability to respond to that. Now, I just want to make the point here that the antibody with the mimetope binding will deplete, but that takes some time by comparison with the therapeutic. And if you go from the right-hand side to the left-hand side, you'll see that we're talking about the dose of antibody injected. The slightly brown bars reflect the dose of Campath, the original antibody injected, and you'll see that as low as 0.01 milligrams will virtually deplete, whereas the mimetope antibody, even 500 micrograms at 24 hours, is only giving us limited depletion. But by 8 days, the depletion of the mimetope antibody is getting greater, and you'll see by 21 days, lymphocytes are now beginning to return. They're being made by, new ones are being replenished, whereas the ability of the mimetope antibody to deplete still continues. If you compare at 0.5 milligrams or 0.25 milligrams, you'll see that there's still depletion going on. So we've changed the kinetics of depletion to make it slower, and that's consistent with generating some capacity for tolerance. Now, I just want to make one point in this slide now, which is you don't have to have every molecule in that preparation being the toleragen to get tolerance. So if we spike our perfect toleragen, the aglycosyl mimetope-containing antibody, with 1% of active antibody, that preparation can still induce tolerance when both are injected together. So even though 1% of the antibody can bind, can give aggregates, can give danger signals, the 99% that's toleragen wins. And that's shown, I can't even see it myself. I think if you see look on the left-hand curve, you'll see at the 5 microgram at the 5 microgram dose, you'll see a very small response compared to what is possible. So substantial tolerance has occurred, even though there's some contamination with the therapeutic. So what I'm trying to say there is that, and that translates actually into having a longer serum half-life on the right-hand side of the therapeutic antibody. So what I'm trying to say there really is that in fact, it doesn't matter if you get a little bit of cell killing, as long as the ratio of the monomer over the therapeutic is advantageous, say over 99%. And just to summarize what I've said this far is that tolerance seems to correlate with long-term depletion as well. I mentioned to you the antibody depletes late, and because the antibody lasts longer and it's not removed, at 21 days you still see very good depletion. If you tolerize to Campath and you then give the therapeutic, if you pre-tolerize, then give the therapeutic, you see that therapeutic lasts for a long time. On the right-hand side in the middle there, pre-tolerization and giving Campath, you get very sustained depletion for a long time. So I just want to conclude in the next few minutes. The stealth version of Campath is non-cell binding, it's not immunogenic and tolerogenic, but that's not absolute, it's a quantitative phenomenon. The antibody to cell binding retains its biological activity, but is immunogenic. So here are the points I want to make, if I can make them. Don't know what's happened here. Next. There are many reasons for immunogenicity, but the main ones we think are to do with creating multimers, aggregates, and generating danger signals that alert the innate immune system. And there might be then the possibility of, if one could avoid those danger signals and minimize the aggregation, there might be generic strategies to tolerize. And of course, some people have pushed the notion of removing all T-cell epitopes and B-cell epitopes from a therapeutic antibody. That's one route, and I'm waiting to see really strong data that that has worked in vivo in a trial. Pre-tolerization could certainly work and could give one very robust tolerance, but probably no pharmaceutical will ever be bothered to do that. And so stealth might. And of course, stealth antibodies, if they work and they have a longer life, you might only be able to give them, like Lemtrada, a single shot and get the benefit of quite sustained depletion. And as I said, I think because we have seen reasonable efficacy in doing this in mice with a totally foreign antibody, we think using humanized antibodies in the human would be far less challenging, and stealth antibodies may even be more effective than the data I've shown you. Now, a few just points at the end, one point at the end. There are various sorts of therapeutic antibodies, and I'd like to distinguish between antibodies that target cells in the blood, which are immediately accessible to danger signals. So if you target cells in the blood, you can imagine lots of antibody immediately binding to those cells, immediately attracting inflammation. But if antibody has to get out of the blood system into a tissue, you could imagine circumstances where, in fact, the blood levels are quite high, and before enough antibody has gone into the tissue to bind and attract danger signals, a tolerance might have been generated. So I think where people might have been using high doses of antibody to tissue antigens, I suspect that there would have been some tolerogenicity going on anyway, which we never, of course, measured. But I think one conclusion here would be, for me, that when we're dealing with antibodies to epithelial targets, it may well be that the stealth technology really has more chance of working really well there than perhaps when we're targeting cells in the blood, where you have immediate access to the cell. So finally, let me just finish by thanking Various people have made important contributions here. There are many people involved, but the preclinical work with mice has been mainly the work of 3 colleagues over many, many years. The clinical studies have been largely led by Alistair Coles and Jeff Hale, and then we are continuing work to try to improve these stealth antibodies, and that's work of Chai Django and Steve Cobol. Thank you.